Hi class, this is a review video for chapters 4 and 5 inside of the cell and the dynamic cell. Let's go ahead and get started. We'll start with cells, how you're able to see them and how they're different from each other. So actually, what is it that makes a cell a cell? Like, there are some requirements. A cell is something that is living, and so the way that we determine something is living is if it is able to metabolize. And so, like, a virus is very small and it has genetic material, right? It has like DNA or RNA, but a virus cannot metabolize. And so that's why we do not consider viruses to be living. So the actual smallest living thing is a cell, and cells are able to metabolize. They have DNA, and that's how that's different from a virus, which is non living. They are diverse. They could be diverse in size, the way they look. Um, and so we're going to kind of go over a lot of those things, more so like what's inside of a cell that makes it different from other types of cells. How you're able to see them. There's two different types of microscopes. So there is the light microscope, which is more of the common conventional one that you'll see in a laboratory. But this one you can see cells that are larger, like a euglena is a protist, and that's a little bit larger, so you could see it with a light microscope. But something really small, like if you really wanted to see something not living like a virus, you would have to use something like an electron microscope, something that is able to see something really, really small. Or if you want to look inside of an organelle, that's really, really small, so you might need something like an electron microscope. So that's the main difference between these, as well as cost. These are much more expensive than a light microscope. All cells do have a boundary or an outer part of it, which is what we call the plasma membrane. And the plasma membrane has a really important function, which is to let certain things in and out of the cell. What it's made out of is what we call the phospholipid bilayer. So it's made out of lipids, and there's also some proteins that are embedded inside of this bilayer. There's a polar head and a non-polar tail. When we say something is polar, that means that it's okay with water. And so the scientific word for that is hydrophilic. So hydrophilic is water loving, or it likes water, and that's also polar. Non-polar is not like water fearing, water hating, and so the scientific term for that is hydrophobic. So let's kind of look at what a phospholipid looks like. So right here is one phospholipid, and so it has the polar head and the non-polar tail. Again, polar means that it's okay with water. It is hydrophilic. Non-polar is water hating, and that is hydrophobic. And so because of the chemistry of how this is with the polar side and the non-polar side, that's why these are oriented in that way. So the heads of the phospholipids are going to be touching water, and the part that does not like water is not going to touch it, so that's why they're oriented on the inside. So the non-polar part is here in the middle, and the polar part is here on the outsides. The same can be said with these proteins. These large purple things are proteins. This part of the protein is polar, and the inner part is also non-polar. So hydrophilic, and then this is hydrophobic, which is non-polar, and then this is polar, hydrophilic. So try to be a little bit more familiar with those words because you'll see those on your test. So non-polar, hydrophobic, polar is hydrophilic, as well as you'll need to remember the phospholipid diagram, why they're oriented in that way. As far as the proteins, their function is to let things in, like larger things. Things like a water molecule is larger, sodium is larger. Something that could pass through the phospholipid bilayer are going to be things like a gas, like CO2 is able to go through these phospholipids, and it doesn't need to go through a protein. But something that is a little bit larger is going to need to be using a protein to go through the phospholipid bilayer. Some proteins don't have a function of just having things pass through. Some are used as markers or for cell signaling. The two types of cells are prokaryotic and eukaryotic. But before we go there, let's start with the cell theory. When we were talking about theories Last time, we established that a theory is something that has been proven. It's not anything to question. This is something that has been tested over and over again, and it is known to be true. So the cell theory is how do we put this? Like we mentioned earlier, so for something to be alive, it has to metabolize. And so all organisms are made of cells, and all cells come from pre-existing cells, meaning that you can't just have sporadic life. Life had to come from pre-existing life. And then it goes into what all cells have in common. They all have a plasma membrane, they have a cytoplasm, and they all have genetic material. We say that they have genetic material specifically because it could be either RNA or DNA. And so to alleviate that it's RNA or DNA, we just say genetic material. The cytoplasm is that jelly inside of the cell that most people don't really know its function, but it has a really important function. One, yes, is kind of like as a to keep things in place because it's a gelatinous material, but the main thing that this does is this is where metabolism happens. This is where ATP is made, and ATP is the energy that the cell needs to be able to do processes. So cytoplasm actually has a really important role that might not have been known to you. So let's go ahead and move forward to the types of cells. There's the prokaryotic and the eukaryotic cell. The main difference between pro and eukaryotic is that prokaryotic cells do not have a nucleus. They have no nucleus, and a eukaryotic cell does have a nucleus. And then we're going to go over more of the differences between them. But this is the main one. Prokaryotic cells, they have no nucleus. Eukaryotic cells have a nucleus. Let's take a look at a prokaryotic cell. One thing about them is that they're really, really small, much smaller than an animal cell, much smaller than a plant cell. They are much smaller than that. And then also we notice that there is no nucleus, but it does have genetic material. So it doesn't have a nucleus, but it has what we call a nucleoid region, and this is how you spell it right here, nucleoid. So no nucleus, but a nucleoid region, and that's where it's able to keep its genetic material. It does have a plasma membrane. Like here, you see right here, this outline right here. It does have a plasma membrane. It does have ribosomes, which are these red dots. It does have a cytoplasm because it does make ATP. And this is a flagella that's used for what we call locomotion, and that's just another word for movement. There's some extra things that you will not find on an animal cell, like the capsule, which is a gelatinous outer surface kind of layer. And animal cells can have this. This is what we call pili, and for an animal cell, we would call it cilia. So, but it does have, you can have these like little hair-like structures, which are also used for locomotion. So main thing about this, it's small, it has no nucleus. Instead of a nucleus, it has a nucleoid region. In comparison, this is an animal cell. It's much larger, much larger, and it has a lot more going on. And so I just wanted you to kind of look at it really quickly before I go to this slide, and then I'll come back to this picture. So eukaryotic cells, they do have a nucleus, and examples of eukaryotic cells are protists, fungi, plants, and animals. And they also have a lot more organelles. So organelles are things like the nucleus, ribosomes, the ER, the endomembrane system, cytoskeleton, cytoplasm, oh, I'm sorry, we said that already. The chloroplast is the one I was looking for. So in an animal cell, you have the nucleus, this big purple thing in the middle. You have the rough ER here, the smooth ER, the Golgi apparatus. There's lysosomes, which are these. It does have some mitochondria, and then it does have a vesicle, centriole, and it has the plasma membrane. There's a few things in this diagram that are missing, but this is just kind of the synopsis, and then we'll go over these organelles. In comparison to the animal cell, you have a plant cell. So we know that this is a plant cell because of, not because this is green and the other one's not green, but some of the giveaways that this is a plant cell is one, this really thick plant cell wall. Another thing is this really huge vacuole in the middle, and then these super big chloroplasts. So those are kind of the three dead giveaways that this is a plant cell. Again, the cell wall, the big vacuole, and the big chloroplasts. Everything else is the same though. It does have a nucleus, rough and smooth ER, Golgi apparatus, mitochondria. So those are just how they look in comparison to each other. In another slide, we're going to mention this because this is on your test. This plasmodesmata. So what it is it's kind of like a channel or a highway. And so this is how cells that are next to each other are able to exchange materials. They could be exchanging proteins, they could exchange food, they could exchange water. And so that's how plants are able to share things amongst cells. So the main function of the nucleus is this is where genetic material is stored. And then another thing that happens inside of the nucleus is that this is where ribosomes are made, specifically in what we call the nucleolus. So this is the nucleus, this big, large thing. And then inside of the nucleus, there's another sphere, and that's the nucleolus. So this is where ribosomes are made. And ribosomes are very important because they are where protein synthesis happens. And so right here it says that protein synthesis. So protein synthesis is just a fancy way of saying that it makes protein. So what will happen is that inside of the nucleolus, it'll make a ribosome, and the ribosome will exit through these nuclear pores. There's like holes right here. It goes out through these nuclear pores. It'll either attach to the rough ER or they'll just kind of be free floating in the cytoplasm. So that's their main function. The ER, the endomembrane system, there's two parts. There's the rough ER and the smooth ER. The rough ER is where you're going to find the ribosomes, but it also does make these transport vesicles to transport ribosomes. And then the smooth ER, the function is to make lipids. There's a picture that I'll show you earlier that kind of shows how they're like actually working together. But so the rough ER, we call it rough because it has all of these ribosomes attached, and the smooth ER has no ribosomes attached. And this is, I think that's the same thing what was on the other slide. So let's go ahead and go to this side. So this is a great diagram. It shows you kind of how they work together. So up here is the nucleus. And then up there would be the nucleolus. It spits out the ribosomes. They either attach to the rough ER or are just free floating in the cytoplasm. And so the rough ER, it says here that it's going to make proteins because it has all of those ribosomes. So it makes proteins and then it puts them in these transport vesicles. And then those vesicles then go to the Golgi apparatus. Once they're at the Golgi apparatus will send them out. And it sends them anywhere to the cell that needs proteins. So the Golgi apparatus, the function is to sort and transport proteins. So Golgi apparatus, sort and transport proteins. The smooth ER makes lipids. The rough ER is going to do protein synthesis and also make these transport vesicles. Another thing that I want to note before moving on is this one, the lysosomes. So what the lysosomes do is they clean up the cell. So if there's anything like extra or even anything that's not functioning anymore, like if a mitochondria stops working, the lysosome has digestive enzymes that it'll go to that non-functioning organelle, whatever it may be, and just break it down and then gets rid of the waste. So that's how it's able to clean up the cell. Vacuoles, the function is to store water and to store food, but know that they're in different types of cells. So you can find them for sure in a plant cell, but you also find them in animal cells. And this is a protist, and they have different functions as well. It's not only for storage, but it could be for regulation. So like this protist, if it's in a salty water condition or if it's in a fresh water that becomes too salty, the way it's able to regulate is with these contractile vacuoles. Some of the two larger structures that we mentioned are the mitochondria and the chloroplast. Let's go into the mitochondria. Mitochondria can be found in both plant cells and animal cells. So yes, plants do need them. And the reason that plants need them is because plants make sugar, which is glucose. So they make their sugar, but they cannot use the sugar as energy like in that state. You cannot just take in sugar and then there's your energy. So what it has to do is it has to move that sugar to the mitochondria, and then the mitochondria is able to convert the sugar into ATP. Once something is in ATP form, then the cell can use it as energy. It cannot use just sugar as energy. That's not how it works. It has to be broken down from sugar to ATP. So that's why plants need a mitochondria. It can't just have a chloroplast. So the process of how it does that, how it takes sugar and turns it into ATP, we call that cellular respiration, where it breaks down carbs, which is the glucose, breaks down glucose to produce ATP. So anything that I have bolded here, you really need to remember that for your test. We mentioned with the chloroplast, that it does make sugar. So the way that it makes the sugar is it's going to use the sun. The sun will hit the photosynthetic pigments, and then it is able to make sugar out of that. Actually, it's like amazing, right, that it can do that. So they use solar energy to make carbs, which is glucose, through the process called photosynthesis. Plants can do this, algae can do this. You don't specifically need a chloroplast to do this. This is not on your test, but you can, if you have photosynthetic pigments, something like some protists do have photosynthetic pigments, also bacteria, they're able to photosynthesize without actually having a chloroplast structure. Something to note that's really, really interesting and also true, it's not like just a thought, but chloroplasts and, I need to put this, and mitochondria, both of them, they have their own DNA. They have their own DNA. The theory is that these were once their own bacteria cell. They were engulfed by a animal cell or a plant cell, and now they just kind of live there forever. And so that's what we call the endosymbiotic theory. But yeah, they do have their own DNA, which is really interesting. There's no other organelles that are like that. So only the chloroplast and the mitochondria have their own DNA. How cells are able to move. We mentioned in the earlier one with the, I think it was a prokaryotic cell, that they had flagella or flagellum. So flagella could be one, cilia is another one. Cilia are the small hair-like looking ones. They have them noted here for you. So cilia are these, and these are the flagella. So they're used for movement, or what we call locomotion. I've got two pictures for you to look at as far as the cell wall in different ways. So the cell wall is something that's really tough. It's really rigid. It helps keep that cell from absorbing too much water and then it like bursting. So that's one of the main functions, is it's like a protection. And then it also has what we call the plasmodesmata that we mentioned earlier. So it's these little channels that the cell is able to send things through. So this is a diagram of how it looks here, and this is the same thing, that it has these two channels. Also in this slide, they just wanted you to remember that cell walls, they're not only in plants, you can have them in fungi and in protists. They're just made of different things. Like for plants, the cell wall is made out of what we call cellulose. For fungi, it's chitin. And so that's how they're different, but they're still what we consider a cell wall. And then we're going to move on to the other parts. This is more talking about energy and enzymes. So where do we get all of our energy from? Like in the whole world, where does the energy come from? It comes from the sun. And so the sun does provide us all that energy, and then it's kind of diverted into different systems. So our biosphere gets its energy from the sun. All of our energy comes from the sun. And there's two basic forms, potential and kinetic. So potential is what we call kind of like a stored energy. And then kinetic energy is this one, energy of motion. So once something is moving, it has kinetic energy. If something is standing still, it has potential energy. So that's how we kind of decipher them. The two energy laws, the first law and the second law of thermodynamics. First law, conservation of energy. Energy cannot be created or destroyed. It can be changed from one form to another. Second law is that energy cannot be changed from one form to another without a loss of usable energy. So the first one is just the flow, that energy is just moving throughout systems. And the second one is that you do lose a little bit of energy, usable energy, when it is transferred. When we're talking about chemical energy, we mentioned earlier, like we're converting glucose into ATP, but what actually is ATP? So ATP is adenosine triphosphate. This is what cells use for energy. So cells cannot just use sugar as energy. It has to convert the sugar into ATP, and then the cell can use it. So cells use ATP to carry out all of their activities, or nearly all of the activities. And so this is what it looks like. It has the adenine, the ribose sugar. So this is a nucleic acid, and then a sugar, and then 3 phosphates right here. And so the way that the actual energy comes from ATP, like where does the energy come from? So you have, this is ATP up here, and then the energy comes from one phosphate group when it's lost. So here you have 3 bonds. When this last bond is broken and this phosphate group leaves, that is when energy comes out. So this is a TP triphosphate. This is a DP diphosphate because there's only 2. So that's where the actual energy comes from when one is lost. So it says ATP releases energy quickly. Breakdown can be easily coupled to an energy requiring reaction. But what I really need you to know is that it comes from this last phosphate group. When that bond is broken and the phosphate is released, that's where the actual energy comes from. And then you just kind of have this cycling of going from ADP to ATP, losing a phosphate group, going back to ATP, gaining a phosphate group, and it's just this cycle. It just keeps doing that, and that's how you get the actual energy. We mentioned the flow of energy, where it comes from the sun. So here the sun will hit the photosynthetic pigments, and then it's able to make a sugar. That sugar goes to the mitochondria. The mitochondria makes ATP, and then that's how you can get energy to use it for cell functions. This will be so this is a little bit harder to, I guess, conceptualize enzymes and how they're able to speed up reactions, chemical reactions. So what an enzyme is, I should probably put this. So what an enzyme is enzyme, I'll just put it here. Enzyme is a protein. So enzyme is a protein. So this blue thing, this is a protein. And so what its function is that a substrate will lock on, they break apart, and it creates a product. And so enzymes are really large molecules, proteins are really big, and so they help reactions speed up because it's able to break down substrates. And they're also really specific, and I think it mentions it somewhere here, that enzymes are really specific. And so this might be a little bit off topic, but it might help you to conceptualize it a little bit. So like enzymes are really important when we're talking about digestion as far as like food. So for most people, if you drink milk, that has a sugar called lactose, right? And that's a sugar found in milk. So if you need to be able to break down that sugar so that you can use it into usable, solid, singular form carbs. Man, this is a little bit off topic. Okay, so lactose is 2 sugars put together, and so once those 2 sugars are put together, you need to break them apart so that your body can actually digest it. It can only digest this if it's in singular form, and then further break that down into ATP. If you have the sugar in this, like where it's just 2, your body's not able to digest it, and you just kind of either poop it out or throw it up. That's just kind of how it works. So when you ingest the lactose sugar, which is 2 sugars put together, you need an enzyme to break it down, and the enzyme is lactase. So if you drink milk and you do not have the lactase enzyme, your body will reject it, and that's either through vomit or throwing up. So those people are lactose intolerant because they don't have this enzyme. They don't have the lactase enzyme to break down the lactose sugar that then can be broken down into your body to make energy. And so that's why enzymes are really important when we're talking about digestion. But the other main thing is that they help speed up a chemical reaction. So what an enzyme has is it has what we call an active site, which is right here. So this is an active site. This is where the substrate locks onto the active site. Once it locks onto the active site, the reaction will occur, and then it spits out these 2 different products. And so there's either the lock and key method or the induced fit. So lock and key is just like that, it fits perfectly. It fits perfect. Induced fit is that it will slightly change the shape to accommodate the substrate. So like if these do not fit perfectly, it'll change the active site shape just slightly though. So those are the 2 different types of active sites, that it's lock and key or induced fit. And let's kind of go into how it is that they are able to speed up a chemical reaction. So this is the rate of the reaction from start to finish. And so which one do you think is going faster? Do you think the red one is going faster or the green one? Let me give you like 2 seconds. So it's the green one, right? This is the long way to go all the way up and then to go down. Whereas you can take the shortcut. So if you are going to use an enzyme is kind of like using a shortcut. So this uses more, this one right here, this one uses more activation energy, whereas this one uses less. So enzymes use less activation energy, and that's how they're able to kind of speed up the process. And then the last part that we're going to go into is movement inside of cells within the plasma membrane, how things can pass in and out of the cell. So there's only 4 more slides left and then you'll be done. So there are 3 ways that they can go through the plasma membrane, which is kind of weird that the chapter has it this way. These slides I would have liked to put in the other chapter, but this is, I don't know, this is how they had it. So there's 3 ways that you can go through the cell membrane. One is passively, actively, or through bulk transport. And so the differences between these 3 is that passive uses no energy. Let me bold this right here. So passive transport, there's no energy needed. It's just able to move through the membrane, and so that's why we kind of call it passive. It's passive, it's able to move through the membrane using no energy. No energy means no ATP. So for active, this one, it does need energy. So active does need energy, which is ATP, and it goes from low to high. So what does that actually mean, right? Passive, it says high to low, and then here it says low to high. So what this means is that it's going to go with the concentration. Oh wait, that's misspelled. Concentration gradient. Let's let it fix it for me. Concentration. There we go. With the concentration gradient, and then this one will be against. So that's like saying this is going with the flow, active transport is going against the flow. So like if you're swimming with the waves, if you're out in the ocean and you're going with the current, that's going to be an example of passive transport. If you're swimming against a wave, then you're going against the concentration gradient, and that's what we call active transport. Bulk transport means that you're moving really big things, and this does need ATP. So this is a type of active transport. Let's make that a little bit smaller. So those are the 3 different types: passive, active, and bulk transport. Passive, no energy. Active needs energy. Bulk transport does need energy as well, with the concentration gradient, against the concentration gradient. Please make sure that you review this, because this will be on your test and it might be a little bit tricky. So if you don't remember these definitions like word for word, I recommend that you practice it, because you're going to get a question and I don't want you to get confused. So make sure that you do review this slide and the definitions. The last thing are going to be solutions. There's 3 types of solutions within cells. There's the isotonic, there's hypotonic, and the hypertonic. So isotonic means that this is happy. The cell is equal, it's happy, like they're just happily shaped, there's nothing happening. That means that there is equal amounts of water on both sides of the cell, meaning outside and inside of the cell. So this is isotonic. The concentration of water is the same on both sides of the membrane. Hypotonic means that there's more water. It says concentration of water outside of the cell is greater than in. So when there's too much water coming inside of the cell is going to get really, really big, and the plant cell will not burst because it has the cell wall to protect it. But an animal cell, or like a red blood cell, does not have a cell wall, and it has the potential to burst. The other word is lyss. So it may lyss or burst if it's in a hypotonic. So hypotonic, a lot of people like to think of a hippo because hippos are big. Hypotonic, hippo, like really big fat cells that have the potential to burst. If it's one of these, plant cells will not burst. Hypertonic, these are going to be things that they're really skinny cells, like they're shriveled, really sad shriveled cells. They're probably in a saltwater solution, and so that's why they're getting really skinny. So the concentration of water outside the cell is less than inside. The cell will lose water, they shrink. Plant cells undergo plasmolysis. Plasmolysis means that the plasma membrane pulls away from the cell wall. So practice these words because these also are going to be on your test. So anything that I have highlighted or bolded for sure is on your exam, and I really want you to kind of review it. And so best of luck on your exam. Please reach out to me if you need me, and I'll talk to you guys later.